So today we have two presentations. Dr. Jeremy Schmit from Kansas State University from the physics department and Dr. Pu-Chen Ke from Monash University, Australia. I also would like to welcome the panelists, Dr. Feng Ding from Clemson and Dr. Jianhan Chen from UMass. Due to the difficulty with the time difference, Dr. Pu-Chen, the second speaker, will not be able to join us live, but we have the recorded presentation from him. So we'll telecast that so you can listen to that. So at the end of both the presentations, we are going to enable every one of you to participate. We can have breakout rooms and have relaxed times as well. So with that, Janidan, it's all yours. Get started.
Thank you, Rams. And I'm Jianhan Chen from UMass Amherst. So it's my great pleasure to introduce the first speaker of today, Jeremy Schmit. He's a friend and a longtime collaborator. So let me just give you a quick background of Jeremy's training and career. And Jeremy obtained his PhD degree from Northwestern University in 1999. Then he went on to UC Santa Barbara and obtained his PhD and studying polymer physics with Phyllis Pincus and Alex Lavin. Then he spent 2 years as a postdoctoral fellow working with Janae Condev at the Brandeis. Then he joined UCSF as a postdoctoral fellow with Kendall from 2007 to 2011. So from 2011, he joined Kansas State University as an assistant professor in the physics department, and where he is now where he's an associate professor now. So Jeremy actually make a few kind of groundbreaking records when he was at Kansas State University. He was the first faculty from physics to obtain support from both NIH and the biotech industry from Amgen. And he probably is also the first physics faculty at Kansas State to renew his R1 from NIH. So Jeremy's research is mainly focused on studying protein phase diagram. And his research has a really unique approach of developing this relatively simple but really insightful microscopic models that allow us to describe this really complex phenomena, including amyloid aggregation that he's going to talk about today. And this kind of model has been also been really, really powerful in helping us understanding liquid phase transition mediated by proteins. So Jeremy, the screen is yours.
All right, thank you very much for that wonderful introduction, Jianhan. It is my pleasure to be here. I first want to stop and thank Rams for the invitation and the opportunity to present, and thank everybody in the audience for taking time out of what is undoubtedly an incredibly busy time of year to attend. Probably not much in the way of introduction is needed for this crowd. This is a talk about amyloid aggregation, and in a Zoominar on proteinopathies. Probably everybody's very familiar with these diseases that all seem to be have this common characteristic of these beta sheet rich protein aggregates. And this really speaks to some level of common principles. There's something in common between these aggregates, despite significant variations in the sequences, and the hope is that there might be difference or commonalities in eventual treatments and mechanisms as well. Now, coming at this from the protein physics point of view, one of the things that really jumps out is that there are just a huge range of timescales that are relevant in studying this problem. So for instance, you're interested in Alzheimer's disease, the number one timescale that you are interested in is, of course, the progression and onset of the disease. And this really shows up out in the decades to even centuries timescale when you're starting to talk about how long it takes for people to show the first symptoms. This, of course, is not a relevant timescale in the laboratory. I have not had the opportunity to tell a grad student that I want them to do a PhD. It'll take 10 to the 9 seconds, but I imagine it doesn't go very well. And so most laboratory investigations tend to happen in much more reasonable timescales, on the order of seconds, hours, minutes, something relatively convenient there. Now, obviously, this is a huge gap in timescales here, but this isn't the only gap that we're dealing with. When we're looking at our primary tool for investigating things going on at the molecular level, that's at least theoretically, this is molecular simulations. And what we see is that there's another gap that opens up in here between the simulation and the in vitro aggregation. And so I see my job as a theoretician to try and plug these two gaps here. This talk here is going to be primarily spent on the short timescale gap. There's other stuff I could talk about here, but in the interest of time, we're going to focus on down here. And when you look at this right off the bat, the presence, the fact that there's a gap here at all is somewhat puzzling to begin with. And the reason for this is the structure that I showed on the previous slide. These aggregates are almost entirely beta sheet. And secondary structure is one of the first things that happens in protein folding. This tends to happen down in the nanosecond timescale. So it's already puzzling that this gap exists at all, and that we haven't been able to just brute force molecular simulations and watch these amyloid fibrils grow. So before we proceed with this, it's probably useful to take a little bit of a diversion here and discuss how normal globular native proteins fold. And one of the key insights of this came from this back of the envelope calculation known as the Leventhal paradox. And what it basically argues is that for a reasonable shortish protein, you've got just this astronomical space of states that you need to sample. And even with some very aggressive estimates on how fast you can switch between the states, you find out that in order to sample this many states, you're just timescales that are many, many, many orders of magnitude longer than what's relevant. Now, of course, the resolution to this paradox is that it's not a random search. It's not an ergodic search over all states. There's an energy landscape that guides proteins to fold to their final state. And this is typically depicted as a funnel shaped landscape, where at the top of the funnel, you have this large space of high entropy states. And then as the native interactions start to set in, it gradually restricts the search space until the proteins reach their final state. And again, this is typically depicted by funnels. Very often it's a smooth funnel, but in reality, we all know that it's a very rugged funnel with lots of traps, incorrect states that need to be broken and then reformed. Now, I want to take a moment to point out here that the fact that we have these funnel-shaped landscape, this is a consequence of evolution. Most random heteropolymer sequences do not have funnel-shaped landscapes. And the fact that globular proteins do is required that these molecules would not be useful if on a physiological timescale if they did. So evolution is going to very, very rapidly push them to something that is funnel-shaped or otherwise just get rid of them. Okay, so with that little bit of a primer, the question is, what does the misfolding landscape look like? What does the amyloid landscape looks like? And again, taking a quick moment to look at the structure of an amyloid fibril, this very naturally suggests something of a simple reaction coordinate. And what that coordinate is basically the alignment of an incoming molecule with this established template that is the rest of the fiber. So often I'll denote this as using what I call the registry variable, which indicates your various alignments, where a registry of 0 indicates perfect alignment where you're perfectly matching your template. And so the natural question here is, how long does it take a molecule to sample all of these alignments? Well, in order to do this, what I did is I mapped this process of a molecule coming down and forming and breaking beta sheet hydrogen bonds with the established template as a one-dimensional random walk. And so we have our fictitious random walker, and it's indicating how many hydrogen bonds I have at a given moment. And over here, it's broken all the bonds and it's detached, and eventually it'll make it all the way back over here and form all the bonds as well. Now, of course, this is not a purely random walk, it's a biased random walk because these hydrogen bonds have a favorable energy. So our walker would rather hop to the left, to the right than it would to the left. And just set your intuition here, the energy scale is roughly that a hop to the right is about 1.5 times more likely than a hop to the left. So at this point, we've reduced the problem to a one-dimensional convection diffusion problem, which can be solved analytically. Continuum, discrete states, there's just all kinds of tools that can be used to do this. And what comes out of this is that we can identify 2 important reaction coordinates and 2 very important timescales that come out of the problem. So the first reaction coordinate is this alignment that I discussed before. Okay, so here I've got a molecule that shifted 2 spots to the left relative to the template. 1 spot to the left, this is perfectly centered on the template. 1 spot to the right, 2 spots to the right. And then within each one of these alignments, the second reaction coordinate is the formation of hydrogen bonds. So this is 1 hydrogen bond, 2 hydrogen bond, 3 hydrogen bonds, 4 hydrogen bonds. Okay, and again, I'm using hydrogen bonds here as a reaction coordinate, and this reaction coordinate accounts for things like side chain interactions and other stuff that's going on there. Okay, so let's talk about timescales. Okay, the formation and breakage of secondary structure hydrogen bonds is incredibly fast. This is happening on the nanosecond time scale. This is that Gaussian peak that showed off on the very left side of that plot I showed, that back of the envelope plot I showed 3 or 4 slides ago. However, in order to shift alignments, these molecules have to break all of their bonds and either form much weaker associations, non-hydrogen bond contacts, or completely dissociate from the fibril to begin with. And this is very slow because it has to break lots of these favorable bonds to do this. And the back of the envelope calculation shows that for a molecule the size of A beta, this should happen on about the millisecond timescale. And of course, A beta is about 40 amino acids long. It's got parallel, anti-parallel registries. And so this millisecond timescale here corresponds to an overall fibril elongation rate on the order of seconds, which is consistent with experiments. Even at this really incredibly coarse-grained level, this model can make predictions. So for instance, we can understand the effect of denaturants and temperatures on fibril elongation rates. In both cases, weakening the bonds here causes an acceleration in the aggregation rate. This is an Arrhenius plot, so we're weakening the bonds, we're heating it in this direction, the bonds weaken, the aggregation rate goes up. And this is a consequence of the fact that most of your time in the aggregation process is trapped in these misaligned states. Okay, so the bottom line here is that unlike protein folding, we have a couple of very good reaction coordinates in order to describe the amyloid formation process. Those reaction coordinates are the number of H bonds that you have and the alignment of your molecule with the established fibril. I should probably say those in the other order. Your alignment and then the number of hydrogen bonds that you have within that alignment. And so we can do a lot of things with these reaction coordinates, and we can understand a lot of what's going on. The next part of the talk, I want to talk to you about how we've used this insight of these reaction coordinates in order to obtain molecular resolution in the elongation process. After that, I'm going to talk a little bit about how we can understand sequence effects here and how sidechain perturbations modify the landscape and allow molecules to either aggregate faster or slower. The third thing, which I'm not going to talk about today, is we've done a lot of work on how amyloid fibrils nucleate. And those are the kinds of things that I'd be happy to field questions about afterwards or by email, but this is something we've done a lot of work on, and I'm not going to talk about it at all today. Okay, so let's talk about molecular resolution. And of course, here what I'm talking about is molecular dynamics simulations. And so let's briefly talk about how protein folding simulations are done. Very often, they're one of the most common techniques these days is Markov state models. And what this is it's taking advantage of the fact that there are a lot of basins, these free energy basins, on the way from going from the unfolded state to the folded state here. So the recipe that tends to be followed here is that the first thing you need to do is identify the basins. Very often this is just some brute force simulations, long sampling times, and you use clustering algorithms to figure out which states are part of the same free energy basin. Then what you want to do is you want to sample the transitions between the basins. For this, you can use enhanced sampling techniques, umbrella sampling. You can put your thumb on the scale and really see what the transition states look like. And then once you sample the transitions and you get some estimates for the kinetic rate constants, then you can assemble a transition matrix and assemble long trajectories here. Now, of course, the problem is that the long timescales of amyloid aggregation make it really difficult to identify the basins in amyloid aggregation. But we've got a shortcut here because we've got a back of the envelope, a pen and paper theory here that's telling us where the deep free energy basins are. And this is when I walked across campus and was very fortunate to find Zhen Han as a molecular dynamics expert on campus. And I said, hey, let's see if we can put our brains together here and figure out how we can use this free energy landscape here in order to jump up several scales in time. Okay, so this is the free energy, or sorry, this state space that we started with. And the way we attacked this problem was a little bit of divide and conquer. And when I say we, I'm primarily talking about Zhen Han and his postdoc, Ziguan. And what they did is they broke this problem into three pieces. The first piece is the beta sheet piece. And this is the piece that's described by the back of the envelope theory here. And it's defining these states in terms of one, this alignment reaction coordinate, and two, the extent of bonding within that reaction coordinate. And so what they did is they went along and did lots of, let's see, it's not going to play unless I do that. They ran lots of targeted molecular dynamics simulations where they watched these molecules in all the different alignments, and they watched these hydrogen bonds break and then reform and break and reformed. And we basically extracted rate constants for every one of these black arrows in every alignment with every pair of residues that you can get. The next thing we did is we looked at, or they, I should say, they looked at these states where the molecule is not in the beta sheets space. And in order to do this, they used sort of standard Markov state model clustering approaches and clustered these and identified the key basins over here. And finally, the third piece of the problem was the diffusive approach of a new molecule to the end of a fiber. And so this is the summary of that work. What it does is it describes the transition of a molecule from the soluble state down to form these in-register hydrogen bonds with the end of the fiber here. And so this purple arrow here indicates the on-pathway set of states. This is the on-pathway set. Now, what they identified is that there's three weakly bound states that do not have beta sheet character that essentially serve as a kinetic hub in this process here. And these determine whether you go down the on-pathway branch or you go onto these two off-pathway branches. One of these off-pathway branches contains about 12, again, non-beta sheet states. And these are very short lifetime states. They can interconvert extremely rapidly. And essentially, these are kinetically irrelevant. On the other hand, this misregistered beta sheet pathway absolutely dominates your assembly times. In fact, what Ziguan observed in his simulations is that the proteins spend about 98% of the time in these off-pathway beta sheets here. And without free parameters, these simulations capture aggregation rates to a factor of about 2 to 4. So in the next slide, what I'd like to do is I've gone through there and I've hunted through Ziguan's trajectories extremely extensively, and I found the most representative trajectory, and I wanted to share that for you right now. This is what it looks like. So we've got a molecule, it diffuses in it, it forms some hydrogen bonds with the end of the fiber, it wiggles around. And the key thing to observe from this is that when it wants to shift registries, what it tends to use is these weak states that are sort of stuck to the side of the fiber in order to hang on while it inchworms over and then grabs on. But it does not slide across the top of the molecule here. It's kind of like Velcro in that sense, and it needs to break the hydrogen bonds before it can transition. So we wanted to assess these simulations here, and in order to do that, we used this smallish fragment of A beta. And we chose this fragment here because it had some interesting mutation effects here. This is the wild type, and it aggregates at some rate. But this experimental group down here tried a couple of hydrophobic mutations on it. This hydrophobic mutation makes it aggregate a little bit faster. A different hydrophobic mutation makes it aggregate still faster than that. But the interesting thing about it is that when you make both hydrophobic mutations together, you get this non-additive effect. And qualitatively, this non-additive effect came out of the simulations. And so we were really interested in understanding what this was coming from. And of course, having these large ensembles of trajectories, we could go in and we could look at this at high resolution, and we could try and understand what it was, what these mutations were doing that would modify the trajectories. Now, looking at the individual bonding rates, the hydrogen bond fluctuations here, we saw that some of these mutations sped up the transitions between the hydrogen bonding states. Other mutations actually slowed things down and caused things to stick. But when we looked at these things in aggregate, we didn't see anything that really could explain these growth rate trends here. And that wasn't too surprising to me. I thought that this was too high a resolution to really see the long scale effects that we were looking for. What seemed a lot more promising was to look at these free energy basins that we identified in the theory and see if we could figure out which ones of these basins had gotten deeper, which ones had gotten shallower. And when we did that, I was left with a much more frustrating conclusion that we couldn't really explain the growth rate behavior even at this scale either. And so it didn't emerge from these registry lifetimes either. And so we wrote what to me was a fairly frustrating paper that said that the net effect of mutations was this gradual accumulation of perturbations over the entire state space. However, things became a lot clearer once I started to think about it in terms of this funnel folding picture here. And so again, we've got these funnel-shaped landscapes that are thought to control the way native proteins fold to their globular states here. But of course, these funnels are, as I mentioned before, a consequence of evolution. And we're not talking about a functional state here where there's going to be an evolutionary pressure to drive it to fold. In fact, we're talking about a pathological state. And if anything, evolution is going to exert a pressure to keep it out of these aggregated states. And so what that means is that we're not looking at something funnel shaped, we're probably looking at something flat and unbiased. What this means is that our trajectories trying to find the aggregated state are random across this space. We're going to undergo some random walk across here. And what this means is that there are no dominant trajectories, there are no dominant transition states. And what you naturally expect from that picture is that the net effect of mutations is this accumulation of positive and negative effects over this entire free energy space. Okay, and so the conclusion from that is that without a dominant pathway, it's necessary to sample this full ensemble of non-native states. And we can do that with this technique where we've done a divide and conquer on the states. Now, one thing that's misleading about this before I move on is that this shows a very flat landscape. And what I mean by a flat landscape is I mean unbiased. There's nothing here that's telling this molecule that it should move to the left as opposed to the right. But this does not mean that it is a smooth landscape. And what you should instead be envisioning is something very rough. And so if you're trapped in this free energy basin here, when you pop up, there's nothing that tells you that you're going to preferentially go this way versus that way. And so this is what this space should look like. So the next thing I want to do is I want to go in and look a little bit more closely at how a sequence affects this landscape and see if we can, back out in resolution a little bit, coarse grain away a lot of the detail of the molecular simulation and get some big picture view of how side chains and sequence are affecting things. So, so the first thing we might do is take a look at two limits here. One limit is the homopolymer limit, where we just have something like a poly Q or something where it's just the same amino acid over and over again. And in this case here, you would expect very little sequence specificity, and you would expect a lot of disorder in the way that these peptides can stack together. On the other hand, we've got molecules like A beta, where NMR and other studies have shown that they have nearly crystalline order here. And so what I want to do here is compare and contrast these two cases here. And so let's start off with the homopolymer limit here. And again, this is actually a relevant system here because the homopolymer poly Q is the core of Huntington protein and Huntington's disease. So this is not just a polymer physicist dream. There's real physiological relevance here. So I'm going to claim that when you have a homopolymer, you're looking at a state space or a landscape of states that roughly looks like this. And so let me calibrate you to what I'm talking about here. The minimum energy state is when the two molecules are stacked directly on top of each other. This gives you the maximum number of bonds between your adjacent amino acids. And if I shift these molecules out of alignments, well, these are two bonds I don't get to form. Again, another two bonds I don't get to form. And then here I'm barely forming any bonds around them. And of course, if I shift them in the other direction, it's symmetric on the other side. So you get with this V-shaped landscape here as a function of the registry here, but I'm going to caution you that while this does look funnel shaped here, this is misleading because I haven't put in the barriers between these states. And if I put those barriers in, it again looks very rugged here. This guy here needs to climb all the way back up to the top before it can transition over and find the native state. Okay, but just looking at this collection of states, or this V-shaped collection of states here, if we compare the expected thermodynamic expectation values of those states, it actually compares reasonably favorably to NMR measurements measured by Patrick Vanderwell's group regarding the misalignments of Huntington protein. The red dots are their NMR experiments, and the blue curve is what you expect with just a standard one-dimensional partition function. You can see that they're off a little bit. Actually, this plot is a little bit misleading because we're all off a lot in the vertical direction, even if we're not off by very much in the horizontal direction. But as soon as you look at the sequences over here on these flanking regions, the C38 and the N17 regions at the N-terminal and the C-terminal ends of the poly Q core, some very, very minor assumptions are all you need to tweak this into really good alignment with the experiments. So I'm not going to talk about that in detail. There's some assumptions that go in there, but the idea is that just the standard partition function actually works surprisingly well. Okay, so now let's add a little bit of sequence heterogeneity here. And I'm going to do this in a theorist way where I do things as simple as possible. And I'm just going to take two types of amino acids, hydrophobic, these are the more sticky ones. I'm going to denote those with black circles. And then the white ones are your less sticky ones. I'm going to call those P for polar. This is Ken Dill's HP model, essentially. So here what I've done is I've concentrated most of the affinity in the center. I've made an aggregation hotspot in the center of this sequence here. And what this is going to do is it's going to perturb the landscape here, and it's going to make it steeper here in the middle. And it's going to help enforce this registry specificity here because now these small mismatches are penalized more strongly because I'm not just breaking a bond, I'm actually breaking a strong bond. Okay. We can do other sequences here. Here I put the hotspots out in the flanking regions. This gives you an even steeper funnel shaped thing here in the middle here. Again, I'm not putting the barriers between these states. So there are these sharp barriers in the middle that I've left out for clarity. The interesting feature that comes out of here is you get these secondary minima on the outside, and this is when, the left hotspot interacts with the right hotspot. And then you can play other games too, with alternating sequences here. We're actually able to do a lot with this model. We could solve the kinetics for these models in closed form, pen and paper, and we got a lot of insight out of that. I'm not going to discuss those right now. What I'd like to talk to you is what happens when we take this model and we apply it to a real sequence like A beta. And to that, when we first did that, we got this surprise here that not only is this thing not funnel shape, this looks like an inverse funnel. In other words, this looks like one of these, horrible, holes in the mini golf course where they put the hole at the top of a hill and you miss by just a little bit, and all of a sudden the golf ball is rolling all the way back down to where you started from here. And that really seems like what's going on here. Now, the force field that we're not using a force field, we're using basically bioinformatic parameters in order to get this. And so this is not very trustworthy at all. But I was really curious as to where this was coming from. And my first guess at the time was that this is just the chemistry principle of like attracts like, and that as you start to shift all these side chains apart from each other, they all get unhappy, and this gives you this sort of inverted funnel landscape here. But the referee on this paper had a really insightful idea. And he said, actually, I know who he is, it is a he, said, you wrote an entire paper here using the HP model here. What if you translated this A beta sequence into the HP language and see what this landscape looks like? And this is what the A beta sequence looks like. And we didn't try different HP models. This is just the first one we tried here. And we translate it to the HP sequence, and you've got one free parameter in this case. You've got to ask how much of your binding energy gets put in the H's and how much gets put in the P's, but we can do that with this constraint that the overall free energy matches the solubility measurements in the system here. And to my great shock, we got something that looks very, very similar to what we got from the much more detailed bioinformatic parameters here. And so to me, what this tells us a story here is that from a variety of methods, our kinetic models, as well as aggregation prediction algorithms, we know that these hotspots of clustered hydrophobic amino acids drive aggregation a lot. They provide a lot of stability here. And I can talk to you about how that looks in a kinetic model as well, although I'm not going to talk about that right now. But it's just important that you have these gaps in the hydrophobicity here, such as these, and you want them sort of scattered through the sequence in sort of a semi-random manner. And what that does for you is it ensures that when these molecules come together and they miss, even if they only miss slightly, what this does is you get lots of mismatches throughout the sequence. And that can give you this sort of order 10 kT perturbation that is enough to reject that misalignment in a physiologically relevant region of time, and then the molecules can come back in. Okay, and so these gaps in the hydrophobicity are really important for providing the specificity that allow you to form ordered fibrils and sort of just disordered aggregates. Okay, so this is the picture here. Again, the argument here is that you have this very rugged but unbiased landscape here. And so what that looks like is this right here. So at the top, it's very unbiased. So there's nothing guiding you to the left versus the right, but it's very rugged because it's got these deep free energy traps. But adding just a few perturbations in the sequence does a lot to flatten the sequence out and make it so that these off-registry ones get rejected in reasonable amounts of time. And this helps it search this space faster and eventually find these tracks, which are the in-register alignments. So with that, I'll put up my summary slides. My 3 bullet points to carry with you are that amyloid aggregation has 2 very good reaction coordinates. The formation and breakage of hydrogen bonds, which happens very fast, and this space of alignments with the fibril template. This is a much slower search. This happens on sort of a millisecond time scale. And because that registry alignment is so slow, amyloid aggregation is very much limited by this search over different alignments with the template. And finally, when you introduce sequence patterning in here, this allows you to rapidly or relatively rapidly reject these misaligned states. If you've got on the order of 10 to the 2 registries, which you do with something with like A beta, you need a discrimination on the order of 10 to the 2 in terms of the binding lifetimes as well. And so 10 to the 2, what you're looking at there, you're looking at about a 5 to 7 KT difference between the in register and out of register states. And that can be explained just with a simple HP model. So with that, I'll put up my acknowledgement slide. I do have a postdoc opening in my group, and if anybody's seen this and likes this way of thinking or know of any theorists that are interested in trying these types of models, I'm looking for applications. I've got a lot more Lego videos. This was sort of my pandemic endeavor, and so feel free to take a look at those on my site. And with that, I'd be happy to take any questions.
Okay, Jeremy, thank you for a wonderful talk. So everyone, please type your questions into the Q&A panel, and I will read the questions from the top as they come in. So Jeremy, the first question is from Patricia, and she was asking, how does the model shown in paper, the PNS paper from 2020, capture the fibril polymorphism?
Right. I don't think we accounted for that. In fact, what we're just doing, I mean, we've got this space of alignments here, and we're asking this question of how quickly does a molecule search this space? And so we are and so polymorphism is going to be, you know, a secondary basin in that search space. And so this is something that 2018 paper discusses fairly extensively there, is that your ability to identify one basin out of multiple possibilities is going to be directly related to your aggregation rate. So if you go to extreme supersaturations, high concentrations, or deep quenches, all of a sudden now you're going to start getting molecules trapped in these out of register states. And this is going to, you know, lead you to polymorphic states, or at least heterogeneity in your fibrils. There's other things of polymorphism that you could get, you know, if you know, you have charges shifting things around. But I think most polymorphism actually happens sort of at the second or the tertiary structure level, where the folds are between the beta sheet alignments. And that doesn't show up in my theory yet at all.
So the next question is from Martin Musko, and he had a quick question. What happens to the off-pathway states when you have a more biased search by including sequence heterogeneity?
Okay, repeat that real quick. What happens to the
off-pathway states when you have a more biased search by including sequence heterogeneity?
Well, again, I mean, the question is you've got two key timescales here. You've got the arrival timescale of new molecules coming to the end of your fiber, and then you've got a sampling timescale, which, you know, how long it takes your molecule to sample these different timescales in order to fall off. And so if your arrival timescale is faster than your sampling timescale, this is when you're going to start to get a lot of defects, heterogeneity, and disordered aggregation there. What you can do with what the sequence does is, again, it smooths out this sample, the landscape here. It allows it to sample this space faster here. And so it decreases, or it increases the time scale for this alignment search, and helps it to keep up with the incoming flux of new molecules.
Okay, so the next question is from Yvette Miller. He said, thank you, Jeremy, for a nice talk. 2 questions. First, can you differentiate between the secondary nucleation to primary nucleation? And the second question is, do you consider only beta sheet in the nucleation? it might be also other nucleation pathways.
Yeah, good question here. Okay, so I haven't talked at all about nucleation in this talk, but the results coming out of the nucleation studies are that, well, okay, so big picture, nucleation is an entropic barrier. In simple colloidal aggregation, you're paying a translational entropy, and small clusters don't have enough bonds in order to pay that translational entropy penalty. When you're talking about amyloid aggregation, again, it's an entropic penalty. One part of the entropy comes from recruiting new molecules from solution, but you have another contribution coming from the conformational entropy cost to straighten your molecules into a beta sheet. And actually, this looks a lot like just a two-dimensional crystal growing with an anisotropic surface tension. You've got translational entropy on one dimension and conformational entropy in the other direction. And once you do that, classical nucleation theory works pretty well. Now, when you're talking about classical nucleation theory, the way heterogeneous nucleation works is it helps you get over one of those barriers. It gives you, when you have an impurity or a defect, it gives you some binding energy that helps you to pay the cost of climbing over that barrier. And I think that's what secondary nucleation is doing, is that the side of the fibril is providing a binding surface that helps to immobilize that first beta strand, so that you're not paying as big of a conformational entropy cost in order to nucleate. And regardless of your nucleation pathway, the barrier looks the same. What things like defects and secondary nucleation do is they provide you a little bit of extra energy to help lower that barrier or help you get it over it more easily. But I think understanding what the barrier looks like is sort of a hard step, and then you can sort of very quickly start to envision what different pathways over it might look like.
Great. So the next question is from Kalina Klein, and she said, reflection talk, do your simulations offer any insight about the influence of beta sheet sequence length on how quickly an energetic minimum can be found?
Yeah, actually, that's pretty straightforward. That doesn't even come from the simulations. That's the back of the envelope theory. And so basically, the problem with a beta sheet is that the peptide backbone looks the same no matter where you are. And so when these things come down, they form these hydrogen backbones, and they very quickly just zip across the top of the fiber. And then in order to shift your alignments, you've got to break all those hydrogen bonds. And so what this says is that the search time is going to scale exponentially with the length of your beta strands. Every time you add a new hydrogen bond there, you're increasing the depth of the kinetic traps there. And so the time scales are going to go up exponentially. Correspondingly, you need to run your experiments at lower and lower concentrations in order to allow it to search that space. The sequence is going to, change that, a little bit, but not by very much. The basic story is that your search time scales exponentially with the length.
Right. So Jeremy, I think along the line, can I ask a follow-up question on that about thinking about longer sequence? So how would the consideration of multiple nucleation site contribute to this kind of prediction of exponential dependence on the sequence length? Okay.
Are you talking about nucleation or elongation here?
Either way, but I'm thinking about peptide can come in and start to sample the registry at multiple spots.
Yeah, I think what's going on is that this is a much longer story here, but nucleation is far more promiscuous than elongation is. And the reason is that conformational entropy penalty means that the beta sheets stay very short in the nucleation stage. You've got these very long disordered tails, and these tails are going to become ordered in the final fibril. But you've got these very large amounts of the molecule that are disordered during nucleation. Once you've nucleated a beta sheet, then it's going to try and assemble a longer strand. And this is when the big sequence perturbations kick in. But nucleation is actually relatively insensitive to sequence just because of the fact that the nucleus is so much the nucleus is so much smaller than the overall size of your molecule.
Yeah. Okay, cool. So we have a lot of questions coming in. So the next question is from Schmit. And the question is that, can you explain how a seed of fibril can dramatically accelerate the in-Basis alignment?
Well, what it does is it helps you get past nucleation, and it helps you get past this conformational entropy barrier there. We also have a nucleation model that explains how you can have these seeding effects. For example, there are experiments showing that if you take A beta and fragments of A beta, they nucleate much faster together, but then the resulting fibrils don't have impurities from the cross species in there. You have the entirely full length fibers and the fragment fibers, and they're separate. And this just, again, it's a consequence of the fact that your nucleus is much smaller than the overall sequence. Nucleation is just more promiscuous than elongation is. That's send me an email. I'm happy to send you a preprint on that.
Right. So the next question is a little confusing. I'm trying to understand the question, but I'm going to just read it verbatim and see if you understand. So okay. The question is from Wahid. The question is that during the alignment process, is there any difference between the two ends of the new chain from the geometrical viewpoint? As you showed in that short MD video, one side of the beta chain was connected to the other chain via a turn.
Yeah, good question. I think it's been shown experimentally that, like, for instance, A beta fibrils grow faster at one end than they do at the other. This doesn't show up at all in the pen and paper theory. I know that Jin Han and his simulations, they'd see different effects based on, because their molecules binding anti-parallel, and they'd see differences based on whether you're binding on this surface or that surface. But no, that the heterogeneity between the two ends of the fiber, I think that's probably going to show up at a level that doesn't show up in the bead and spring model. Anything more I said than that would just be falling into the category of wild speculation.
So, okay, so there are three more questions. So I'm going to read the question from Camilo La Rosa first. And the question was, nice talk. Amyloidogenic proteins have a biological role. Under physiological conditions, they do not aggregate. In pathological conditions, they aggregate. This means that the aggregation is due to changed environmental conditions. So in your talk, you did not mention the role of solvent in aggregation. What do you think about the role of solvent in aggregation?
Yeah, so of course, so what the solvent's going to do is it's going to effectively perturb these binding energies, these hydrogen. When I say hydrogen bonding energy, I mean the hydrogen bond plus the side chains and these types of interactions there. And I'm not sure I agree that physiologically your conditions are changing all that much, but it certainly makes a big difference in vitro experiments. Physiologically, it's probably more proteostasis machinery that's breaking down. But again, that's a guess. But yeah, it can be incorporated in this process here. And in fact, at the very beginning of the talk, when I showed those temperature and denature independent plots, that's what I was doing, is I was accounting for the effect of the solvent on the net binding energy. And so that can all be accounted for here. And it affects those solvent conditions. What they do is they change both the binding energy of the specific state and the non-specific states at the same time. And that balance can be exploited to either make things grow faster or more robustly or whatever.
Great, Jeremy. So the last two questions I think are from the same anonymous attendee. And the question is regarding the plasticity of incoming monomer. So I'm not sure I completely understand the question. So there are two questions asked, but I think they're the same. Maybe I'll read the second one. Do you consider the plasticity for the mutation in incoming monomer?
Oh, I don't understand that.
Yeah, I'm not sure I understand that either.
So what I first thought in terms of plasticity is how mobile do the molecules need to be when they, and I would argue that that if molecules tend to form their hairpin structures in solution before hitting the fiber, that's actually inhibitory toward aggregation. And the reason is that that hairpin constrains the mobility that they need to peel off if they get it wrong. And they're going to get it wrong the vast majority of the time. So what you want is highly mobile, highly disordered molecules, because those are the ones that are going to be able to search the space more rapidly. Whereas if they, the more they fold, the more they constrain their dynamical degrees of freedom, the deeper those traps are and the harder it is going to be to break out. I'm not sure that's what the question was about. That doesn't connect to mutations at all. So,
yeah. So for this particular attendee, I think if you want to reach out to Jeremy, just shooting an email, I'm sure he will be happy to talk to you more.
Absolutely.
Yeah, and let's thank Jeremy for a wonderful talk again.
Thank you for having me.
Thank you very much, Jeremy, for the great talk, and thank you, Jonathan. Next speaker is Dr. Pu-Chen Ke. Dr. Feng Ding is going to introduce him.
Yeah, hi. So it's first we had a great talk, which is amazing. So next I'm going to introduce Professor Pu-Chen Ke. So Pu-Chen is currently a deputy director of the Nanomedicine Center at the Great Bay Area National Institute for Nanotechnology.
Go ahead, sorry.
Oh, okay. And also he's an adjunct professor at Monash University. Before he joined this institute in Guangzhou, he was a signature project leader at Australian Research Council Center of Excellence for Nanobioscience and Technology, which is located at Monash Institute of Pharmaceutical Science at Monash University. And he also was a distinguished visiting scientist at the Australian National Lab. And before that, actually, he was a tenured associate professor at Clemson University. Over the years, he already authored more than 160 journal papers on protein corona, amyloid aggregation and mitigation. And also he has a lot of work on nanomedicine and nanotoxicology and environmental science. One of his early work, he established about like nanoplasticity to the environment, which has been highly cited in the field. His research has been well-funded, both from NSF, NIH, and EPA when he was a tenured professor at Clemson, and also from Australian government, the Ministry of Science and Technology of China, which is the current center he's working on. Dr. Ke is also well decorated with awards. He got the award of inaugural Supervision Excellence Award of the Australian Research Council at Monash, and also he was Faculty Achievement Award at Clemson, and also a Career Award from NSF. And he's also serving in multiple editorial board, including Biophysical Chemistry and ACS Nanoscience AU. So with that, I will leave the screen to Dr. Ke's pre-recorded video.
Pu-Chen Ke here in Guangzhou, in the very southern part of China. I'm 12 hours ahead of the time in Michigan, so I'm pre-recording this video just in case. And thank you, Professor Ramamosi, for the kind invitation. And today I'm presenting my topic, which might be slightly more applied than the other topics at the seminar series. My topic has two components. One is on the amyloid aggregation, and the other side is on the use of nanomedicine against the aggregation and toxicity of amyloid proteins. There are over 30 human diseases that are classified as amyloid diseases. For all of these human disorders, they are categorized by the presence of some amyloid proteins, such as A-beta and tau associated with Alzheimer's, alpha-synuclein associated with Parkinson's, and human islet amyloid polypeptide associated with type 2 diabetes, as well as PRP with prion diseases and others. And for all these amyloid proteins or peptides, they start typically from the monomeric disordered form, and then they gradually aggregate into the sometimes quite toxic oligomeric species, and to protofibrils, and to amyloid fibrils, and eventually to plaques. And all of these species can interact with the cell. For example, they can impact on the fluidity of the cell membrane. They can drill pores in the membrane through the deposition of the so-called beta barrels, one of the many forms of the oligomers. And they can extract lipids through hydrophobic interaction, especially with the amyloid fibrils, and they can then induce the stress on the ER, production of reactive oxygen species. They can damage mitochondria in ATP production, and they can also damage the nucleus on this apoptosis. And so there's plenty of space for biophysics, as you can tell, in terms of the structure, the aggregation, a conformational changes of the amyloid proteins and peptides. And there's also a vast space for the use of nanomedicines against many processes involved in the pathologies of these amyloid diseases. Since the early 90s, there have been steady efforts dedicated to the inhibition of amyloid aggregation and the toxicity, heavily influenced by this amyloid cascade hypothesis. The use of peptidomimetics inspired by the amyloidogenic sequence of A-beta and IPP, for example, the use of antibodies against the presence of toxic amyloids and oligomers, and the use of synthetic and natural small molecules, including polyphenols, and the use of nanoparticles and their various biocomposites from the 2007. Basically targeting the various species of the amyloid proteins and peptides from the monomeric form to the amyloid plaque form. In terms of physics, the aggregation of amyloid proteins into the final form is pretty much a random walk on the free energy landscape. It starts from this monomers, and then it goes to this beta sheet rich or alpha helical rich oligomers. And then it evolves into this amyloid fibrils occupying the NH minima. The purpose of amyloid aggregation inhibition is to massage this free energy landscape by using nanoparticles, for example, to tweak the surface of the NH landscape so that we can avoid the accumulation of the toxic oligomeric and fibrillar species. as this field involves the use of nanoparticles and proteins and peptides. So it's quite natural to evoke a comparison between this amyloid protein corona, so to speak, on nanoparticles, and the conventional protein corona that is a key concept in nanomedicine. There are some similarities and differences between these two concepts. One is that for the conventional protein corona, the proteins are plasma proteins and intracellular proteins mostly, and they are functional, so they're not pathogenic in nature. And the accumulation of proteins or absorption of proteins on the surface of nanoparticles tends to be fairly rapid, from milliseconds for the soft corona to hours for this hard corona. For amyloid protein corona nanoparticles, these proteins are pathogenic in nature, and they also have a high affinity for each other, so they can aggregate. But they have a relatively lower affinity for the nanoparticle surface. So this formation of the amyloid protein corona nanoparticle tends to be fairly slow, on the order of hours to days to weeks or months, depending on the in vitro or in vivo conditions. So there are quite some differences and some similarities between these two concepts, or these two fields of nanobio interactions. Since 2007, Sarahlin's group demonstrating the use of polymeric nanoparticles against A-beta aggregation, there has been a lot of developments in the field in the use of polymeric and condensed nanoparticles against the aggregation of a range of amyloid proteins from IPP to A-beta to tau, as well as others. The idea of this practice is just to promote the protein nanoparticle interaction to overwhelm the peptide-peptide or protein-protein interaction that can lead to the production of non-toxic species or off-pathway species. Utilizing the non-specific forces of hydrophobic interaction, hydrogen bonding, electrostatic interaction, etc., that are quite common to nanoparticle protein interactions, including the protein corona concept. In addition to this dominant amyloid cascade hypothesis, there are other hypotheses available. One is the gut microbiome. It has been increasingly realized from the past decade that the gut bacteria play a rather significant roles in shaping up the health of the brain in the pathologies of cancers. So it's quite essential to understand how the gut microbes might impact on the aggregation and toxicity of amyloid proteins. In this particular study, we used a bacterial amyloid protein called FABC, which is associated with the extracellular matrix, the amyloid matrix of Pseudomonas that is implicated in gut inflammation. And as you can see in the THT curve on the far left, the A-beta aggregation, the blue curve, and the A-beta aggregation in the presence of FABC seeds, the orange curve. So this, in the presence of FABC seeds, the nucleation phase of A-beta disappeared, the elongation phase was shortened, and the saturation phase was elevated, so indicating a promotive role of FABC in the A-beta aggregation. And this is in vivo data. The A-beta was injected to the brain of a zebrafish. Over 1 week, the amyloids and the plaques of A-beta started to accumulate as indicated by Congo red fluorescence. And in the presence of Fc, it did not take it did not need to take 1 week, only 2 days. The accumulation of A beta plus and amyloids was very evident in the as indicated by Congo red. In terms of behavior, as you can see, with the injection of A beta to the brain of a zebrafish by 1 week, the movement frequency was much impaired compared to this to the A beta control right here. And in the presence of FCCs, that stage was reached in 2 days instead of 1 week in comparison with A beta. So again, it's quite accelerating process by the seeding of FAPC. And in terms of the distance traveled, swam by the zebrafish for A beta by 1 week, they could, zebrafish barely could swim. And in the presence of FC by 2 days, and that stage was reached again. So this implicated the accelerating role of bacteria amyloid protein FAPC in promoting the aggregation and toxicity of amyloid beta. There are other schools of thought, so to speak, and here I just present a few. We proposed that amyloid fibrils are colloid-like, so they can easily pick up plasma proteins as well as intracellular proteins to impact on the protease status. And in a severe case, they can cause collapse of protease status in the intracellular space. And here, this is a pancreatic beta cell exposed to the 3 species of human islet amyloid polypeptide associated with type 2 diabetes. This is the monomer, the oligomer, and the amyloid fibril. The oligomer, as we know, is the most toxic species, and it caused a severe dysregulation of protein expression in the pancreatic beta cell. And we introduced a graphene quantum dot, which is a 0-dimension nanostructure. And the nanostructure was able to recover protein dysregulation as indicated by proteomics. So mass spec data is shown here. And in addition, in recent years, there has been a hypothesis that viral or bacterial infection and the induced inflammation could be responsible for the complications of Alzheimer's disease, for example. And interestingly, in the current pandemic, has been literature showing that there is increased correlation or infection rate among the Alzheimer's patients in getting infected by the SARS-CoV-2 coronavirus in comparison with the general population. So that's quite some intriguing data showing there might be some connection between viral bacterial infection and the pathology of Alzheimer's. Over the past 7 years, we have developed a range of nano strategies against the aggregation and toxicity of amyloid proteins. On the left, here we used a sonicated carbon nanotube, multiple carbon nanotube. They were fairly short, a few hundred nanometers in length, and they were coated with the amyloid of a milk protein called beta-lactoglobulin. Beta-lactoglobulin is a major component of the milk we drink, and they can form amyloid under the acidic condition at a low, basically a low pH and a high temperature. And then we sonicated the amyloids of beta-lactoglobulin to so-called seeds, and we used the beta sheets to stack with the toxic human islet amyloid peptide, especially the protofibrils and fibrils, and to some extent the beta sheet bridge oligomers as well. And that was quite effective in suppressing the toxicity of IPP in zebrafish, as indicated by the recovered hatching and development of the animal. On the right, we developed another new protein called beta-casein and grafted on iron oxide nanoparticles via polymer handle. And this particular inhibitor was effective against the aggregation toxicity of all three amyloid proteins of alpha-synuclein, A-beta, and IPP. And we used both zebrafish and mouse models to support our in vitro data. And here with mouse data, the accumulation of A-beta in the brain of the animal, as indicated by the antibody 811 for the A-beta oligomers. You see this bright dots here. And with the co-injection of beta-casein iron oxide nanoparticles to the brain with A-beta, the occurrence of A-beta oligomers was quite negligible, as indicated by the immunohistochemistry. On the right, we used Western blots to show the suppressed expression of the biomarkers for inflammation, for autophagy and apoptosis, with the co-presence of A-beta and beta-casein iron oxide nanoparticles in comparison with the elevated expression of those biomarkers by A-beta, demonstrating that this particular nanoparticle is quite a universal inhibitor against a range of amyloidogenesis. In this recent study, we injected beta-casein coated gold nanoparticles to the heart of a zebrafish, injected A-beta to the brain of the fish, and the nanoparticles were let circulate through the bloodstream to reach the brain. And in the presence, with the injection of A-beta, as you can see, after 1 month, the fish started to show significant Alzheimer's-like symptom with very reduced movement. And that was recovered by the co-injection of beta-casein gold nanoparticles to the heart of the fish, as you can see by the much increased trajectories of the animal. And in this cognitive function assay, we connected electrodes to arena number 2 of this chamber. A zebrafish with the injection of A-beta was not able to distinguish the left and right-hand side. And as you can see, the distribution on both sides seems to be quite comparable. And in the presence of beta-casein gold nanoparticles, the zebrafish were very well aware of the danger associated with arena number 2. Then they stayed on the left-hand side. So that showed improved cognitive function of the animal with this particular nanomedicine, beta-casein coated gold nanoparticles. At the time, we were not very clear, or we did not explore further on the mechanism of the blood-brain barrier translocation of this gold nanoparticles. And this year, we performed a rather comprehensive study trying to understand how gold nanoparticles can go through the endothelial layer or barrier, including the blood-brain barrier, using experimental methods coupled with computer simulations. So this particular slide, I'm showing only the numerical calculations on the interaction between gold nanoparticles and this weak adhering junction proteins lining up the endothelial layer, and that is for the vasculature and also for the blood-brain barrier, for example. And here we showed with the increased ratio of gold nanoparticles to weak adhering proteins from 0 to 0.75, from black to this blue curve, the rupture force for the weak adhering dimer was reduced from 10.6 piconewton to somewhere under 9 piconewton. So indicating that the gold nanoparticles were indeed effective in disrupting the integrity of weak adhering junction to open up this endothelial paracellular pathway. And in panel F and the panel G, we showed that if the gold nanoparticles were more localized, meaning the gold nanoparticles were more hydrophobic, if they were more clumping, they tend to be more effective in breaking apart the weak adherent junctions as compared to the more uniformly distributing gold nanoparticles or more hydrophilic or amphiphilic gold nanoparticles, as shown down the bottom here. So it's quite interesting to understand how the nanomedicine inhibitors against amyloid protein aggregation toxicity from this biophysical perspective. There are many questions to be answered and to be understood. In the future, we will be exploring some of these directions. The consideration of anti-inflammation, antibacterial, antiviral, and the high blood-brain barrier translocation capacities for the inhibitors we design. The consideration of both primary and secondary nucleation in the amyloid doses of proteins and peptides. The incorporation of omics and machine learning for understanding the accumulating data in the literature on the inhibition of amyloidogenesis, and the consideration of multiple proteinopathies instead of just one A beta for alpha-synuclein, for tau, for example. How can we combine all those together to paint a more comprehensive picture, and also the use of better, more accurate animal models to mimic the symptoms of the amyloid diseases. So there are many questions to be understood. And without going further, I would like to thank my collaborators in the US, in Europe, and in Australia, and here in China, and also thank my previous and current students and postdocs, as well as the postdocs of my major collaborators, and thank the funding agencies, and thank you for your attention.
So we cannot take questions, obviously. So
yeah, I see there's one question. I can briefly maybe help answer it because we had some actually joint work. So from Christian Graysinger to Pu-Chen Ke, is there evidence that, for example, A beta and FAP-C can meet? Normally, the gut lumen where FAP-C resides on bacteria is quite well separated from neurons. So this is actually a very interesting question. So what happened is in the literature, they're already showing data that those FAP-C fibrils can cross the barrier into the circulation. So especially when the barrier is being weakened due to inflammation and all other conditions. And so we know that A beta, it's definitely can in the circulation. So they can meet, for example, in the circulation. There are also a lot of evidence suggests that inflammation can also cause like the even blood-brain barrier being leaky, so that the bacteria actually can causing infection in the brain, and where I think those can meet. So basically, it's in healthy patient people probably they don't, or very rarely they do, but some cases probably they do. Yeah, and
So,
yeah, go ahead.
did you guys find the intact bacteria in the brain, or just the fragmented molecular fragments, molecular components you saw in the brain?
Okay, so in this particular work, because we are biophysicists, right, and he is a physicist also, so we didn't really to have all these samples from patient, but we actually in the zebrafish we actually did inject, so mimicking this model, like injecting the FAPC seed in the stomach, in the blood of the zebra, like a fish, and then we have A beta in the brain, so that we actually can see that those FAPC seed, those sonicated FAPC fibro actually can get into the brain. So, and we also have another actually submitted paper showing that the FAPC indeed can cross using this called paracellular pathway, crossing the endocellular barrier by basically weaken the VE-catering dimer and be able to causing the leakage of the endocellular cells. So in this sense, we have some not in vitro kind of evidence showing that those can cross the barrier, mimicking nanoparticles, basically, those fibroseeds.
Are you comfortable answering the questions from the Q&A from Aiton Lerner and Melachini have questions?
Okay, let me see. So thank you for the wonderful talk. For the gold nanoparticle zebrafish study, what's the optimal size of the gold nanoparticle? Yes, so we found that in our simulation, we can only deal with smaller ones, but experimentally we found that the larger ones, like the bigger, like for example, like 70 nanometer cannot cross, but smaller one, like a 30 nanometer and lower, like 10 nanometer size nanoparticles were able to causing the endocellular leakage. Yeah. And from Eaton Lerner, if these nanoparticles cross the blood-brain barrier, and if they bind specific disease biomarker in the brain, is it possible that these bound nanoparticles will cross the blood-brain barrier back to the blood? In short, can these biomarker bound nanoparticles be used for biomedical diagnosis? That's a very interesting question. So we actually had a recent work showing that the corona, basically the protein coating the nanoparticle can dramatically reduce the ability to be able to cross the endocellular cells. So those reduce, but still some of them can cross. So when the protein fully cover the nanoparticles, the ability for them to cross is reduced. So probably the efficiency of being to see this probably is very rare, I would say. Okay, another question is from Giuseppe Melaccini. Albumin is known to bind A beta assembly in plasma and cerebral CSF. Does albumin interfere with nanoparticles? I think that so that's the whole the field of protein corona. The studies actually those coating of albumin definitely will interfere, change the properties of the nanoparticles. So it's actually a very a kind of answered actually intensive studies trying to understand the effect of protein coronas on all kinds of design nanoparticles, whether they will interfere or screen the design functionality. And also maybe those nanoparticles, the coronas, when they could also causing like a gain of functions, which could lead to toxicities. Anyway, so those are, I think, are still unanswered questions to be addressed.
Thank you very much, Wang. Thanks to Dr. Pu-Chen Ke for sharing his exciting discoveries. Thanks, Jeremy, for a nice talk. Really learned a lot. Great work.